Yes. So are we recording? Yes, we are.
Yeah.
Okay, perfect. Excellent. So I'm gonna, start things and then probably people will start joining while I'm still talking. So that's fine. Welcome everybody. We are in the second guest for our second summer of the series. We are trying to create a space during the summer where students, postdocs, PIs can interact and talk about the things that we are interested in, specifically when it comes to retinal physiology, retinal biology, development, disease, heavy focus on disease, of course. Our last edition last year was very successful. This year we kicked things off with Zheng Jiang last month, and now we are hosting today Ryoji Amamoto, which I will talk about him in a moment. And next month is going to be our third guest, so that will be in August. So stay tuned for that. Um, Let me introduce the speaker for today. Um, Ryoji graduated from the University of Illinois, and then he was a Fulbright Fellow for 1 year in Switzerland. I imagine that's the best chocolate you can get. Um, then he went on to Harvard to do his PhD in biomedical sciences with Paola Arlotta on a project about axolotls, which I think
That's
that
right.
should be fascinating. Maybe we can talk about that later on
Yeah.
a different day. And then, of course, the thing that brought me to his research and reading his papers is his work with Connie Setko. He was a postdoc with her. Connie, of course, is a very well-known name in retinal biology, a very famous researcher. And we were studying what happens to the inner retina when the photoreceptors die and the role of retinoic acid in that process of inner retinal remodeling. And in parallel, basically, Ryoji and Connie were basically looking at what happens to the photoreceptors themselves and the role of retinoic acid in that area. So basically, we are like looking at two sides of the same coin, if you will, and that is why I think his work is really fascinating. Is very important for my work and my lab. But I think that most people here will all find it very interesting. Today, Ryoji will talk about what he's been doing as a PI. He started his own lab in 2023 in Massachusetts Eye and Ear, which is the ophthalmology department of Harvard Medical Center, something like that.
Yep.
Yeah, and he's going to talk about retinitis pigmentosa and the mechanisms of cell death in RP. So, with that being said, please take it away.
Share. Okay. Uh, yeah. Can you? I have to. Nope.
You can share. I
Okay.
think you
Now I should be
can.
sure. Yep.
No?
I should be able to. Can you see that?
Yep. Perfect.
Is that good?
So, yep. Perfect.
Okay. So yeah, thanks, Michael, for the invitation. Michael said, this is kind of informal, so if you have any questions, feel free to ask, whenever you want. Okay, so today we're going to be discussing the mechanisms of secondary cone degeneration in like 99% in mouse models of retinitis pigmentosa. So, like Michael said, I was in Switzerland for a year, and this is a picture that I took when I was in Switzerland. Most of you, probably all of you, can see this perfectly clear picture of beautiful Switzerland. Now, if you are a patient with retinitis pigmentosa, you are born with the mutations that is in many different genes. And what happens is that maybe during late childhood, early adolescence, you start to have night vision issues. So you have a hard time seeing at night. And after a couple of years, could even be a couple of decades, it's quite variable. You will start to have dark spots in the mid periphery where you can't see as well, even in daylight color vision. And then that progresses over time into a tunnel vision. And for some patients, they lose their vision completely. So at the cellular level, which is what we're interested in, what is going on? So like I said, retinitis pigmentosa, or RP, is is caused by mutations in genes that are expressed, generally speaking, in rods only, although some of them are expressed in both rods and cones, or RPE. And there is a first wave of degeneration, which affects the rods, and that is why you have night blindness. And in mice or in human, 95 to 97% of photoreceptors are rods. So you can imagine these cones, which don't even express the gene that is mutated oftentimes. They are suffering because of these neighboring rods that are degenerating. Because of that, there's a second wave of degeneration where the cones, they start to lose their outer segments. And then after a while, the cells will degenerate as well. So if you can imagine that, from birth to, you have this period of rod degeneration followed by cone degeneration. There are several therapeutic options out there, most of them not great, or they are still in development. So if you catch the rod degeneration early on, then you can either have low vision aids, so maybe like wearing sunglasses and things like that, so that the degeneration is slower, or you can also have gene-dependent gene therapy. And most of these that are in clinical trials right now are gene augmentation therapy, where you have a recessive retinitis pigmentosa, you have a mutated gene, and you can just use AAV to put in the wild type copy. And that's been successful. You can also imagine the other side where pretty much all rods are dead and pretty much all cones are dead. So that would be a late stage. People are, again, working on things like optogenetics to regain vision, or even in the future, you might have cell replacement therapy. And there might be also a point of intervention where you have the rods that have degenerated, but you still have cones, and your daylight color vision is, it's deteriorating, but it's still there. In which case, what I'll be talking about would be the gene agnostic gene therapy. So, like I said, gene therapy has been successful, at least in terms of FDA approval. At least one case has been successful, with many on the way, we think, where this is a therapy called Luxturna. It was the first FDA approved gene therapy. And it's, theoretically speaking, it's simple. You're replacing a wild type copy of RPE65, or in this case, it's back into the RPE cells. And it's pretty crazy, these patients, they can go from not being able to see to seeing, faces and obstacles within a couple of months, which is fantastic. So you might be asking, okay, well, why are we even working on this disease? It seems like it's already solved in terms of therapeutics. So there are about 2 million patients worldwide, about 1 in 4,000. And these are the different genes. In total, about 69 genes that have been identified to cause retinitis pigmentosa. So it's very, very heterogeneous. 30% of patients still don't even know what the causal mutation is. You can imagine, so Luxturna, the therapy that I just described, is for this one little sliver of RPE65 patients. So you can imagine that 99% of patients to this day, there is no therapy for them. So again, big pharmas, biotechs are tackling this issue kind of one by one. But at the end of the day, there are dominant cases where you can't just put in a wild type copy, or some of the genes are too big for AAV. So there are a lot of issues, and a lot of them are just way too rare for big pharmas. So what we are hoping is that in terms of rods and rod degeneration, it could be gene 1 or gene 2 or gene X that is causing the degeneration. But at the end of the day, they are converging upon cone degeneration. And cone degeneration is the cause of the loss of daylight color vision, which as humans, we require for, day-to-day function. So we thought, okay, maybe we could treat more patients, maybe all patients with RP if we can tackle cone degeneration. So for that, we are developing or trying to develop a gene agnostic therapy for retinitis pigmentosa. So then it comes to the question kind of for the whole for this topic. Okay, why are these cones degenerating once rods die? So what I want to talk about today is about the vasculature. So if you look at the vasculature in the retina, and this is kind of flipped upside down to what I usually show, but you have the superficial layer here near the ganglion cells, and you have the intermediate layer here in the IPL, and then in the OPL, which are next to the ONL where photoreceptors are, you have this deep capillary plexus. And what I'll be showing you for most of the time will be the deep capillary plexus blood vessels. And these blood vessels are important for nutrient exchange within the retina. And then, of course, you also have the choroid, which also gives nutrients to the cells in the ONL as well. Now, it's very important in the brain or in the retina, which are very susceptible to different insults, that the blood doesn't just escape randomly and the contents of the blood. So, the brain has the blood-brain barrier. The retina has a blood-retina barrier. Here, I'm going to be talking about the BRB or the blood-retina barrier, so that the contents of the blood stay within the blood, unless it's something that the retina needs. So it's composed of tight junctions. It's of endothelial cells. It's also composed of cells like pericytes and Mueller glia and feet to make sure that only certain material gets transported from the inside of the blood to the outside. So that's the inner blood-retina barrier. And then there's the outer blood-retina barrier, which is formed by the RPE cells and the Bruch's membrane. So in our lab, we use mouse models for retinitis pigmentosa. And we use different mouse models, but a lot of it, we discover a lot of the things in RD1 mice, which is a fast degeneration mouse line. In this mouse model, rods start degenerating around P10. And by P20, pretty much all rods have been degenerated. At which point, cones start degenerating, starting around P20. And the reason why I got interested in the vasculature was, and this has been described previously, if you look at P15 in the RD1 mouse, and this is a flat mount of and it's stained with CD31, which stains the blood vessels. You see the blood vessels in the deep plexus. But again, P15 to P20, there is a massive loss of rods. And there's also this vascular regression or pruning that occurs between P15 and P20, where there are far fewer blood vessels in these mice.
Ryoji, sorry, this is the outer retina facing up or the inner retina? Which side are we looking at?
This is looking at like the photoreceptor side. And then we are using a confocal to look at just the deep plexus. Does that make sense?
Yep, sure.
Okay.
Thanks.
And compare that to, for example, CD1 wild type mice that have no degeneration. You can see that in the deep plexus, there's a lot of vasculature. So there's this vascular pruning that's going on. And then what we wanted to see was, okay, we wanted to see if there was any kind of leakage. So at P27, either in CD1 wild type mice or in RD1 mice, we do a tail vein injection of a tracer called sulfo-NHS biotin. And then you wait 20 minutes so that it can circulate. And then you sac and harvest the retinas. And what we started seeing, if you look at CD31, so this is the blood vessels, or the streptavidin. So this is a fluorescent streptavidin that binds to the biotin that was injected, so the tracer. So this kind of shows you where the tracers are. If you look in a flat mount, I saw that there were a lot of regions where the streptavidin staining seemed to not overlap totally with CD31. So, if the tracer was 100% basically not leaky, then the CD31 and the streptavidin should co-localize completely. But in these regions where it's red, but not green, these are the regions where streptavidin, or like biotin that you injected, is leaking out of the blood vessels. So we found this leakage that's occurring at P27 in RD1 mice. And this coincides with the timing of cone degeneration. And when the interesting thing that I thought was also, if you look at cone arrestin staining, so CAR here, cone arrestin, at P27, a lot of the cones in the center have degenerated. And the center is where the leakage was happening. So I thought this was an interesting in terms of spatial correlation. And you can also find, little bits and pieces here where, if you still have cones around, you can find these regions where you have this leakage and the exact region where cones are degenerating, and it seemed to overlap very well. So again, still correlation, but we thought this was interesting. So we thought, okay, maybe this has something to do with cone degeneration. If you look into a section, so this is cone arrestin staining, looking at the cones in the O and L. This is wild type, so no degeneration, no leakage. You can see that the blood vessels are staying within the OPL. This is a deep plexus here. But in the RD1 mouse, where there is degeneration, you have this one layer left of O and L, which are mostly just surviving surviving cones. There are regions like this where biotin that we injected is leaking out, and it's not just biotin. You can also look at things like mouse IgG, which you can just use like a secondary antibody to test if you just see the blood vessels or some of the IgG are leaking out. And sure enough, endogenous material is leaking out as well. So in terms of the temporal correlation, we also, looked at the number of central cones that are surviving. So if you look at P21, a lot of the central cones are still remaining. And then it's very quick degeneration. By P24, a lot of the cones have degenerated, and then P27. And this was similar in terms of looking at the leakage area as well, where P21, there's very little leakage. But by P24, we are starting to see a lot of leakage, and then it kind of continues from then on. So there's a spatial and temporal correlation between cone degeneration and the leakage. This is not totally new. People have found that if you look at, for example, albumin staining, so albumin should generally stay within the blood. But if you look at these black spots, so there's not much of it in a normal human retina, but in a retina of an RP patient, there are these signs of leakage of albumin within the retinal parenchyma. And there are also papers, for example, this is an RD10 mouse where they looked at different tracers and they were able to see leakage as well. But when I was starting this, people had kind of known about this. We had shown it, and it just kind of remained mostly like a correlation. So we wanted to really see if there's any causation between the leakage and secondary cone degeneration. So that gets us to, okay, how do we treat the blood leakages in the retina? So, well, one of the first things we wanted to see was, okay, well, there must be a reason for the leakage. So one of the things that we looked at was VEGF-A expression. And this is again RD1 mouse at P27. On the left here is a no probe control, and then on the right is with the probe. And what we saw was that it's not so much that there's VEGF-A expression just like upregulated everywhere, but we saw these little pockets where VEGF-A seems to be expressed it seems to be more upregulated. And we're in the retina field, and if you talk about leakages, one thing comes to mind, which is anti-VEGF. So with diseases like wet AMD, it's caused by, or one of the symptoms is abnormal vessel growth due to increased VEGF signaling. And luckily for these patients, we do have treatments. One of them is shown here, Eylea, which is an anti-VEGF, and people get injection of this once every 2 months, 3 months to stop the leakages from happening and stop the abnormal vessel growth from happening. And we thought, okay, well, this might be a candidate to stop the leakage in RP. So our design was pretty simple. At P20, we would do, in RD1 mice, we would try an intravitreal injection of anti-VEGF. And then at P27, which is like kind of the height of leakage, we would check for leakage. And in another cohort, P40, which is kind of when we usually look at cone degeneration, we'll do cone quantification. So if you do an injection here, we're just injecting PBS. And then we also do 20 minutes before euthanasia, tail vein injection of sulfo and HS biotin, which is the tracer. There are regions highlighted in arrows here. There are regions where there is leakage. But when we injected anti-VEGF, what was encouraging was that these leakages seem to almost disappear completely. It was very little leakage in many of the retinas we were looking at. And this is the quantification here. So we thought, okay, this is a good candidate for stopping the leakage in the retina. And so what happens with the cones? So this is now at P40 in RD1 mice with a P19 intravitreal injection. With PBS, again, we have cones degenerating in the center. But with the anti-VEGF, we saw a more uniform cone distribution as well as more cones surviving in the center. Quantified here, if you look at the central cones, which was, again, I wouldn't say that it's like going back to the wild type numbers, but it was still a significant increase in the number of cones that are surviving in the center when we stop the leakage using anti-VEGF. One of the more striking things was this thing called a crater. We've known about these things called craters, which are shown here for a long time now. Nobody really knows what they are, but if you look at, for example, glutamine synthetase to look at Mueller glia, you can see these spots, which again, we are calling craters. And these overlap, if you look at cone arrestin staining, of pretty much a zone of 0 cones within these things. And so we call these craters because there are no cones in there. And when we used the anti-VEGF, what was surprising was that these craters were almost totally gone. You can see the quantification. This was probably the more significant result, where the craters are completely gone, which makes me think that these craters might be due to the leakage, and it kind of looks like the leakage spots. So now it could be that the craters are causing the leakage. We don't really know, but at least spatially, there's probably a correlation. So right now we have this working model where in the case where you have intact blood retina barrier, again, you have choroid and you have the 3 intraretinal vascular plexus. Green here are the cones, and then here in the white are the rods. Now there's no leakage here, but once rods degenerate, you can imagine that there's that the cones are now pushed up against the deep plexus, which start leaking, and the leakage here might be what is causing the, or at least contributing to the secondary cones, secondary cone degeneration. Okay, doing 5 for time. Any questions about that part?
I have a bunch of questions. I mean, the idea, no, because when we look at the other side, when we look at the inner retina, we see the effects of remodeling. We model that using a dye that is called YOPRO1, and it's
Yeah.
absorbed in pericytes, but it's also absorbed in ganglion cells. We also see a patches of expression. So overall, the RD1 is a lot more permeant to these dyes that are usually impermeant. And
Right.
ganglion cells, bipolar cells do not, in a wild type retina, they don't load this dye, they don't absorb the dye. And the RD1 does it a lot. 50% of the cells load the dye versus maybe 5% in the wild type. But that 50% is also in patches. It's also in specific areas. You can find an
Like,
area in the RD1 that is completely perfect and
so
doesn't
like
have
these
the other.
craters.
Right. So, I mean,
Interesting.
he's from the other side, of course, but
Right.
I mean, I think this is really fascinating. One quick question. When you look at RD1P60 here and you're still finding cones, I mean, isn't that too late? Are there any cones left in an RD1 by
So
P60?
so this one's P60. The other one's P40.
Okay.
If you look at P60 cones, you still see it in the periphery, the far periphery, which I can talk
Okay.
about. But in the center here, there's like hardly anything.
I see.
So this might be like a remnant, maybe. But yes, you'll definitely still see it in the far periphery. The other thing that's interesting, based on what you just said from the ganglion side, that you see patches, is also that RPE. If you look at the RPE, you also see RPE like the morphology of the RPE gets disorganized, and that seems to happen in these patches as well. So I'm thinking there's something there.
Right.
These like columns.
Like it's a vertical, right? A vertical thing
Exactly,
across the
vertical
retina.
columns.
Right.
Right. Okay, so let me go to the next part, which again still has to do with the blood vessels, so people can ask questions later too. So, like Michael said, my postdoc work was looking at retinoic acid signaling, and here I wanted to use the retinoic acid signaling pathway as kind of a molecular toggle to investigate the cause of secondary cone degeneration, and I'll explain what I mean by that. So this is something that is already published, but if you look at the rare lac-Z mouse, so this is a reporter mouse for retinoic acid signaling. And if you look for lac-Z, so these blue precipitates here, we found that the blue precipitates are in the far periphery. And the lac-Z, which again, it's another name for beta-gal here, in green here, again, it's in the far periphery, and it seems to overlap very well at P40 with where the cones are surviving long-term. So this was interesting to us because, in particular, we wanted to know why these peripheral cones are surviving long-term. And if you look at the cell type that the beta-gal is expressed in, so where retinoic acid signaling is active, we found it mostly in Mueller glia. And this is just summarizing what we had found. But if you imagine these red dots as individual cones, and the red, the cones in the far periphery are surviving, especially dorsally, and that's when retinoic acid signaling is normal. When we have the loss of function, where we can knock out the synthesis enzyme called ALDH1A1 or retinoic acid, and if we knock that out specifically in Mueller glia, the cones, even in the far periphery, which were supposed to survive, are degenerating. And on the flip side, with the gain of function, we can electroporate a constitutively active form of retinolic acid receptor where you can activate the signaling pathway, and within that patch, cones survive long-term. And you can do this in the central retina. So again, this is kind of like the molecular toggle that I'm talking about. We can kill cones, we can help them survive. So now we have this, model where Mueller glia in the periphery with retinolic acid signaling are somehow helping the cones survive longer, where in other areas where retinolic acid signaling is not active, the cones degenerate. So what was interesting from the bat when I was looking at the blood vessels, so this is at like P27, and you look at the CD31 staining for all the blood vessels. So again, these blood vessels have pruned. So only, these ones are still there in the deep plexus. But what I noticed was that these areas in like the far periphery, they seem to be lacking any kind of blood vessels in the deep plexus. And because I was working on retinoic acid as well, I knew that this was probably where retinoic acid signaling was active. So here are the regions where retinoic acid signaling is active. This is the beta gel, and it matched very well. It's an anti-correlation. Wherever there's LAC-Z signal, wherever there's retinoic acid signaling active, there were hardly any blood vessels in the deep plexus, which was peculiar for us. And then so we decided to test this working model where in the red here, you have retinoic acid signaling, and in the middle, you don't. You start out with blood vessels everywhere. And then after rods degenerate, you have remodeling of the blood vessels where you now only have the blood vessels in the center. And in the far periphery, you don't have any more blood vessels, at least in the deep plexus. And that is also where cones survive longer. And it could be because in the center, you have the blood vessels still, which can leak. And in the far periphery, you don't have blood vessels anymore, so it can't really leak. And that's how maybe the cones are surviving long-term. So this was a hypothesis that we had that we wanted to test. So again, going back to the retinoic acid signaling as a tool, so we can use, so this is an RD1. So here I'm just showing you the CAC-CRE-ER with ALDH1A1, 2, and 3. So this is a synthesis enzyme for retinoic acid. So flox. So we can get rid of retinoic acid signaling in these mice. Give tamoxifen at P10 and then look at the retinas at P60. If you do that and look at the retina for ALDH1A1 expression, it pretty much goes to 0. Again, this is something we've shown already in a previous publication. And if you look at the whole retina, again, clones in the far periphery are surviving long-term. But with the CAC-CRE-ER, so now the synthesis enzyme is gone, retinoic acid signaling is gone, even the cones in the far periphery, they start degenerating. So now our hypothesis would say that, okay, well, now if you don't have RA, what happens to the blood vessels? Again, here, you have blood vessels in the center where there is no retinoic acid signaling. So now let's get rid of retinoic acid signaling in the far periphery. What happens to the blood vessels? So here we have RD1, ALDH1A1, so just the one, flox at P40. So if you don't have CRE, so this is kind of like wild type control. Again, in the dorsal side, you don't have any blood vessels. But with the Mueller glia specific CRE, now we start to see a lot of blood vessels that were, it could have been like regenerated, but likely it like never went away. It was never pruned in the first place. So if you quantify that, it goes from almost no blood vessels in the periphery to you have blood vessels in the periphery now. Right now, we don't have evidence to know if these are leaky or not, but that's something that we're testing right now. And then now the the opposite experiment. So I'm just going to take you through what we do for the activation. So if you don't have retinoic acid signaling, retinoic acid molecule, retinoic acid receptor is not on. So it's actually a repressor for downstream gene transcription. When you have the ligand, retinoic acid receptor turns on the downstream transcription. The problem is that if you want to, let's say you just like inject retinoic acid because you want to turn on the pathway, it's kind of difficult because the first thing that it turns on is this gene called CYP26A1, which is an enzyme to degrade retinoic acid. So there's this very strong negative feedback loop. So what we decided to do was we overexpressed by electroporation a receptor, so retinoic acid receptor, fused to VP16, which is a transactivator, and now we can activate this gene even without the ligand being present. So it doesn't matter if there's CYP26A1 or not. And so we do the electroporation at P0 with this CAG RA alpha VP16 construct into RD1 with the retinoic acid reporter and look at P40. So again, this is something we've shown before. If you look at the top here, which is the retinal flat mount, here's the BFP positive region, and this is a close-up of it. So this is the electroporated patch. Beta gal staining, so retinoic acid signaling, is now active in this patch. And if you look at cones, again, in the cones in this patch, it's surviving long-term. This is an image from the publication, but I wanted to also show a couple more images where here's the electroporation patch, and here's another animal, and hopefully you can appreciate that if you look at cones here or here, it's surviving within that electroporation patch. So now, going back to the blood vessels, we have this way to activate the signaling pathway. So what happens to the blood vessels? So here's a control where we just put in BFP, just the electroporation control. So here's the BFP, and here's the CD31 blood vessels. You can see that the blood vessels are still very much there. Now, if you put in the RA alpha VP16 to activate the signaling pathway, now we have this electroporation patch where most of the blood vessels in the deep plexus are totally wiped out or likely pruned. And this is, quantified here. So it almost goes to 0. So now with both of the loss of function of RA and the gain of function, we now know that, okay, RA signaling seems to have a role in two different things. It can help cones survive, and it can also regress or prune blood vessels. It could be that there are two parallel pathways, have nothing to do with each other. But we wanted to know, maybe RA signaling is helping cones survive because it is regressing and pruning the blood vessels, which it would have leaked from otherwise. So to test this, what we wanted to do, and this gets kind of complicated, so stay with me. What we wanted to do was an epistasis experiment where we will activate RA signaling by electroporation of RA alpha VP16. And then usually this will lead to vessel regression. But instead, we're also going to overexpress VEGF, which is the master regulator of angiogenesis. So now, if our hypothesis, if the mechanism we're thinking of is correct, VEGF is going to override any type of vessel regression phenotype of retinoic acid signaling. And then we're going to see if cones survive still, or do they now die? So again, pretty complicated. I'll take you, through it. So this is an RD1. We're looking at P40 retinas, P0 electroporation with CAG BFP, so to mark where the electroporation is, CAL-NL VEGF-A. So CAL-NL is so this is kind of like LOX, stop LOX VEGF-A. So it's a Cre-dependent VEGF-A. So without Cre, it's not on. And then here is a CAG RA alpha VP16. So this is not dependent on Cre expression. and also sulfo NHS biotin to mark the leakages. So now I'm showing you the control where we don't have any Cre. So these animals have the RA alpha VP16. So in this patch, RA signaling is active, but VEGF-A is not there. So here's the BFB expression. Biotin, there's not much leakage of any sort. And it's kind of hard to see here, but hopefully you can kind of see this like linear part where here in this patch area, cones can survive long term, whereas here, there's not much survival. Comparatively, if you now have the cat-Cre, so now VEGF-A is on and RA signaling is on in the electroporated patch. So you have this electroporated patch, and as we would have expected, there is now a lot of leakage. And what was interesting was that within the electroporation patch, the leakage seems to be like in the periphery of the patch. We don't really know why that is, but so that's why I'm showing you this this dotted lines here for where the leakage is. And if you now look at the patch here again, hopefully you can see that, within this electroporated patch, now within this leakage area, cones are not surviving as well as compared to the outside of the leakage area. So we quantified all of this. Again, it's kind of complex. So this is with or without cat-Cre. So this is, and with RA alpha VP16. And we're now looking at the blood vessel length. So again, you could imagine that if you have VEGF-A, because you have Cre here, now you have a lot more blood vessels, which is what we would expect. Same with the leakage. We used to not see a lot of leakage, but now there's a lot of leakage happening when you overexpress VEGF-A. And if you look at cones, so again, here in the first two, we don't even have the RA signaling on. So now we're just looking at the effect of VEGF-A. Here, we're looking at the number of cones, and there doesn't seem to be much of a difference if you just overexpress VEGF-A. But if you look here, now we turn on retinoic acid signaling, and cones survive longer. Now, in this case, we don't have any VEGF-A expression. But when now we overexpress VEGF-A, the number of cones decrease. This indicates that this VEGF-A overexpression and the induction of local angiogenesis kind of gets rid of the cone survival effect of retinoic acid signaling. So again, a lot of these are kind of suggestive still in terms of evidence, so we're still working on some kinks out. But just to summarize, we're here in the red is where retinoic acid signaling is active and where retinoic acid signaling is inactive in the center. The blood vessels, at this point, the rods are still there and the blood vessels are still there. Upon rod degeneration, blood vessels remodel, and now it's only in the center, at least for the deep plexus blood vessels. And what I've shown you is that these blood vessels that remain in the center, they start leaking, and they likely contribute to secondary cone degeneration. Whereas in the far periphery here, the cones might be surviving because the blood vessels are gone, and now it can't leak anymore. So this is kind of the working model that we're working with right now. So with that, I want to thank everybody in my lab. So Sarah used to be in Michael's lab, and she's been a fantastic lab technician who contributed to this project, as well as Joe Bunker, who's a postdoc in the lab, who's spearheading a lot of this work. And also everybody else in the lab, as well as the OGI, which is the institute that I'm part of, as well as Chenghua Gu, who's kind of the blood retina barrier guru, as well as the SEPCO lab for their support and the funding as well. Thank you.
Excellent. Thank you. Thank you, Ryoji. This was really, really great. I open the floor for questions. Please Unmute yourself if you want to ask something. Okay. Do we have anyone? Okay, I'll go first to break the ice. So
Yeah.
let me understand something. You are using CAG. I know this sounds like a technical question, but it's not because
Yes.
you talked about the importance of retinoic acid activation in Mueller glia cells,
Yes.
but then you're using the CAG promoter. Walk me through that. Is CAG for Mueller glia cells or is everywhere or
So yeah, so if you electroporate postnatally with a plasmid with CAG, you're gonna hit postnatal cells. So that would be Mueller glia, rods, some amacrine cells, very few, and also bipolar cells. You're not gonna hit other cell types. And what I mean, I didn't really get into it here, but when we're using CAG for a lot of these experiments, in terms of for example, like the cone survival experiments, we've also used the Mueller glia specific
Mhm.
promoter, and you still see the effect. So we think so. We use CAG sometimes, but it can also be a Mueller glia specific promoter as well.
Okay. And the activator, so I also work with VP16, with RAR alpha VP16, and we saw
Yeah.
something very interesting. On one hand, when we looked at the cells becoming more permeable and opening these pores, it worked amazingly. So you infect the retina from the intravitreal side with a virus, you deliver RAR alpha VP16, and they
Yes.
become very permeant. But they did not become hyperactive, which is what we were after. And then, I realized this is RAR alpha. So it's RAR alpha that is getting constitutively active.
Yes.
But we have also RAR beta and RAR gamma, and we have
Yeah.
the RXRs, the
Yeah.
orphan retinoid receptors. So how do you think the activation of RAR alpha by VP16 is affecting the other
Yeah.
isoforms in the system?
Yeah. Good question. Yeah, like you said, RAR alpha is in pretty much like all the cell types. The beta is in some subset, and there's some other ones. I don't really know what I haven't looked too closely into the transcription factor binding motifs of RA alpha alpha versus beta, but my my naive assumption is that when we overexpress RA alpha VP16, it might also turn on the genes that are activated by the beta as well. I don't
Mhm.
have any uh evidence for that, but just because it's kind of an artificial system, so we're just like blasting the system. So
Right.
I can imagine that it's not necessarily off target necessarily, but just not as on target, I guess. So that's what I would assume.
Right. I must imagine it might be really complicated to electroporate mice at P0, the day they are born. You electroporate their eyes. I
That's
mean,
what we
how
do.
does
Yeah.
that work?
It's once you get good at it, it takes a minute or two
Oh,
for
really?
so it's
Oh,
very easy. Yeah.
oh, awesome. Okay.
Yeah.
Okay. More questions? People in the audience? Anyone? Nope. Okay, we're shy today.
No
So,
problem.
okay. Excellent. Ryoji, this was amazing. I thank you so much for coming over, virtually coming over
Yeah.
and delivering this talk. I hope very much to see you in person next Arvo or
Yes.
any other time. This was fantastic. I'm happy that Sarah is doing well in your lab. Um, and thank you so much. Thank you so much
Yep.
for joining us.
And
And this lecture will be available on YouTube later in the week. Thank you, everybody, for joining us, and see you all.
thank you, everyone.
Bye. Bye. Bye.